what we got to learn about is how transcription is controlled and translation. You see, your DNA has about 40,000 genes on it. That means any cell can make 40,000 different proteins. If you're a cell, do you want to be making all 40,000 all the time?
No, sounds like a bad idea.
That would be very wasteful. This is the DNA in the nucleus. It has instructions for making all of it. But each cell, like your liver cells, only want to make certain enzymes. And your stomach cells make different enzymes. And your brain cells make different enzymes. So each different type of cell is only using a small part of the whole code. Does this make sense? Yeah. Some of y'all aren't listening. Hey, do you know who Larry King is?
He died.
He just died. He's a famous interviewer. He interviewed thousands of people. And his famous quote is, "I never learned anything by talking." Isn't that a great quote? He never learned anything by talking. He learned things by listening. Listening to people. That was his job. So some of y'all need to get good at listening.
Yeah, is there a class for that?
It takes practice. It's like shooting free throws. At first you don't make any. At first it's hard for you to listen. And then when you work on it, you really pay attention, you get better at it. At some point you'll become a good listener.
Did you know he was in the Bee movie as an animated?
What was he?
He was the
Which
Bee? I think he was Larry King.
Larry King the Bee. All right. So, how this works is, how do we control whether the gene is transcribed? Remember transcription. You got to know what transcription is. That's when RNA polymerase attaches to the promoter and then opens up the DNA and adds RNA nucleotides to make mRNA. You guys remember what transcription is?
Yeah.
Thomas, you seem like you're not paying attention here. RNA polymerase will attach to the promoter and then transcribe the gene. And then the mRNA will go to the ribosome and make the protein. Well, that can be controlled. And we learned about last chapter how there's transcription factors that have to be in place to control it. There is a section on the gene of a prokaryote called the operator. It's called the operator, and it's right in front of the promoter. And we're in a prokaryote now, so it doesn't have a nucleus. That's why I erased the nucleus. And there's an area where a protein can attach. And the protein in this case is called a repressor protein. Here it is. A repressor protein. And this repressor protein can attach at the operator.
It's
like a puzzle piece, that's right. And where is this repressor protein produced? It's produced at another site on the DNA. Let me make this gene curl around like this. There's another site on the DNA that codes for making the repressor protein. And it can be way away from this gene. And it has a name, and it's slipping my mind. It's called the regulator gene. The regulator gene. There's a regulator gene somewhere else where, again, the RNA will attach. It's It's got its own promoter and its own terminator. The RNA attaches here and transcribes the regulator gene, and the repressor protein gets made in the ribosome. And the repressor protein floats around the cell, la-di-da-di-da, and then it sticks to the operator. And that turns the gene off. The gene is in the off position. Because this RNA polymerase cannot attach to the promoter here, the repressor protein's in the way. And so this will never be transcribed. This is in the off position.
So, how many of our genes have the operator?
Almost all the genes are off. Only a few are on in each cell. Out of the 40,000 genes you have in a cell, maybe only 400 or 500 of them are on at any one time, which is 1 or 2%. Yeah.
Do they all have like an operator part on it?
In prokaryotes, it seems that they all have an operator.
But some just don't sign to stop it.
The repressor protein that binds to the operator can be removed. I'm going to show you how that happens. That's how we turn it on. Now, this whole thing is called an Operon System. The Operon System consists of a regulator gene that produces a repressor protein that sticks to the operator. The whole idea of this control mechanism is called the Operon System. The Operon System. And the one the book talks about, there's two the book talks about. The lac operon is one, and another one's called the trip operon. I'll talk about both briefly, but I like the lac operon the best. You ever heard that milk can spoil?
Yes.
It can spoil milk. It smells bad, doesn't it? You know why? Bacteria are growing in it.
Yummy.
If you get some bacteria in your milk, in about a week, they'll have multiplied enough where they're all in the milk and they're producing waste gases, and it makes it smell really bad. And it ruins the milk. It'll make you sick, too, because you'll be drinking a lot of bacteria. They'll get in your gut, and they'll cause massive sickness, diarrhea and stuff. Don't drink spoiled milk. Now, if a bacteria finds itself in milk, it will want to be able to digest milk sugar, which is called lactose.
Are those baby lactose magnets?
These are lactose magnets.
Oh my goodness.
You like that? I made those with my lactose magnet making machine.
That sounds like something hand in hand.
Now these genes, listen, Chaz, are you paying attention back there?
I am reading.
This stands for three, right here are three genes that will make proteins that will break down lactose. But it's in the off position.
So,
How do we turn it on? If the bacteria finds itself in some milk, lactose will diffuse into the cell. And one of the lactose molecules will stick to the repressor protein.
That's incredible.
And it will change the shape of the whole protein so it no longer sticks to the operator. And it'll fall off. And now the gene is on. And the RNA polymerase can stick to the promoter. It can transcribe these genes. An mRNA will be made that goes to the ribosome and makes proteins. And you know what those proteins do? They break down lactose. The enzyme, one of the enzymes is called lactase. And it comes over here and it attaches to the lactose and gobbles it up. And it breaks it. It actually doesn't eat it. It breaks it into smaller units. Does anyone know what lactose is made of? It's made of two sugars. Glucose and galactose. Very nice. And so the lactase breaks it into glucose and galactose. Do we want lactase to be built by the ribosome? if we're not in milk sugar, if the bacteria is not in milk, why would you make lactose? You would only make lactose if you happen to be floating around in milk, right? So once all the lactose is removed, it'll even remove the lactose that's stuck to the repressor protein. It'll come up here and get that one too. And then this will change shape back into the shape it had before, and it will bind to the operator and turn off the gene.
So it just does this until there's no lactose left.
It does it until there's no lactose left.
Wait, so if you're lactose intolerant, the gene is never turned on?
No, because this is in prokaryotes.
Oh.
So in you, if you're lactose intolerant, and even if you're not, you may become lactose intolerant as an adult, by the way. You might be able to handle lactose now, and you may not be able to when you're an adult. What happens is you make a version of this lactase enzyme in your digestive system. And people who are lactose intolerant don't make it.
Okay.
So they can't break down lactose. And then the lactose the sugar's not digested, and it gets in your large intestine, where there's a lot of bacteria that will eat it and produce waste gases. And so people who are lactose intolerant, they don't digest the sugar, and it gets in their large intestine, and those bacteria eat it and produce waste gases, and it gives you really bad gas. And you're farting a lot. And the gas can build up so quickly that it blows up your large intestine.
Oh.
And you die of internal bleeding.
That sounds like a balloon.
That's what the blood's supposed to
One of those partial balloons.
Kind of sounds.
So that's basically the lac operon. Does that make sense? Would you like to see video footage?
Can we pump some milk into someone like this?
The exploding large intestine.
I don't have any video footage of that, but you probably wouldn't want to see it.
Bro.
Is anyone like this intolerant in here? Because we're gonna pump you with some milk.
Fine, cold.
Usually,
I absolutely love teaching. I just wish I had more time to
hand
work
feed
on
him
muscles.
peanuts.
he doesn't love food.
The tryptophan operon, TRIP operon for short,
Oh, wait.
is a bacterial system for regulating the synthesis of the amino acid
Hold
tryptophan.
on.
The operon controls
We want the lac operon.
Watch us recording.
Here we go.
Mike loves one of these ones. This is so exciting. I hope this person's good.
Virtual cell.
Heck yeah,
Yo.
the dramatic use of
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon, beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA.
Those are ribosomes.
Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
What do you think about that?
Pretty neat.
Did you know you can zoom by pushing up?
Ooh. Whoa.
Okay, so read. I didn't quite finish. I want to talk about the trip operon. I'll talk about that tomorrow. It's trippy. And you never know. It could be a quiz over, I don't know, I think it's 1.16, 1 and 2.
Oh, and 2. Okay.
Yeah.